Even though cells are the smallest living thing, it is the basic unit of life. Even you initially started as one single cell. Then you as one single cell divides, make more of yourself, and eventually build you as a whole. Cells are the structural and functional unit of life. How well you can function depends on how well your cells are. If something in your cells go bad, then you don't have a good functioning cell, right? That contributes to not properly functioning tissue, organ system, and of course, a not properly functioning you at the end. What you see here is a recording of real cells in the process of making more of themselves. They are dividing. They look kind of cool, right? It's kind of hard to believe that these moving globs of things are actually, they can make up you. There are many different types of cells in your body. We have skin cells, brain cells, muscle cells, and many more. All of these cells look different from each other and have different functions. Cells' characteristics, such as the way they look, for example, dictates their function. For example, sperms have long tails that help them swim, versus skin cells, they like to attach closely to each other and have many layers on top of each other to help with protection purposes. Here is a close-up of a cell. The three main components of a cell is the plasma membrane, nucleus, and the cytoplasm. The plasma membrane wraps around the entire cell and acts as a protective wall that protects everything inside of the cell, kind of like our skin protecting all organs under it. The cytoplasm, which is in between the plasma membrane and the nucleus, and it has a lot of different organelles inside of it. And organelles, each one of them has different function. Kind of like under our skin, we have different organs that do different things for our body. We also have the nucleus, which is this guy here, right? So with the nucleus, it contains our DNA that has all the information on how to build us. Did you know that all of our cells in our body actually have the same DNA? It doesn't matter if that is a skin cell, brain cells, or muscle cells. They all have the same DNA. Then what exactly makes skin cell different from brain cells? Well, that's because DNA has genes, and these genes can be turned on or off. For example, to be a heart cell, you then need to turn off gene, you need to turn on gene 1, 2, 3, for example. And to be a brain cell, then you need to turn on gene 4, 5, 6 while keeping gene 1, 2, 3 off. Let's talk about the first part of the cell, the plasma membrane. Remember how I said structures dictate function? As mentioned, the plasma membrane acts as a physical barrier. It holds the cell together. How the plasma membrane can be such a barrier is because it's composed of phospholipids. A phospholipid has a hydrophilic head and hydrophobic tail. Phobia means fear of, kind of like, you have phobia for spiders, for example. And hydro means water. If you combine the words, hydrophobic means fear of water. The tail of the phospholipid here is afraid of water. It doesn't want to interact with water. But since the head of this phospholipid is actually hydrophilic, means it actually love water, then these phospholipids form a great solution where the head actually touches the water while the tail hides away. And as you can see here, so let's say out here, that's the external environment where there's a whole bunch of water outside, and that's the head. The head is being exposed to water while the tails are hidden. Another thing that you can see from the plasma membrane is that there are some chains of carbohydrates or sugars. We call these glycolics. So let's erase this. These guys here, these are basically chains of carbohydrates, or the glycolics. They serve as signaling molecules that cells use to communicate with each other. This is like the signal that your cells can recognize if this is actually part of you, your own cell, or an invader cell. If your body doesn't recognize these signals, these cells will then be attacked and get killed. This is kind of like when someone has an organ transplant, but their body rejects it and lead to medical complication. Another special characteristic of the plasma membrane is selective permeability. This means only certain things can come in and out of the cell. The plasma membrane has channels and gates, which are these blue things that you see here. And they regulate what material can come in and out of the cell. Now, let's talk a bit more about selective permeability and how the cell control what can come in and out. Things can come in and out of the cell passively or actively. Passive transport refers to things that can pass the plasma membrane without using any energy, moving from high concentration to low concentration. As you can see from this beaker here, at the bottom of the beaker, it's pretty concentrated, right? Because it has lots more solute compared to everywhere else, which is just water. Whenever you have something in a group or concentrated like that, it will always want to disperse out to areas that doesn't have much solute until you have similar amount of solute throughout. Let's go back. This is a very important concept, as we will use this concept in the later chapters as well, because this is how things move around your body. Things always want to move from high concentration to low concentration. This is considered as things this is considered as things that are moving with the concentration gradient, which is from high to low concentration. It's kind of like, when you have too many candy and you got to give them out to people who don't have the candy until everyone have the same amount of candy. This is what we normally call the act of sharing. However, if things want to move against the concentration gradient, which is from low concentration to high concentration, this would be considered as active transport and would require the use of ATP. It's kind of like you have a lot of candy already, but you want more candy. To get more candy, you need to put in the work, which is ATP. Passive transport that we talked about earlier that involve moving materials from high concentration to low concentration is considered as diffusion. There are a few types of diffusion that we want to look at. We have simple diffusion that refers to very small molecules like oxygen and carbon dioxide that can squeeze their way through the plasma membrane without any assistance, though it still has to go from high concentration to low concentration, going with the concentration gradient. Facilitated diffusion refers to larger molecules that can only go through the plasma membrane with assistance. In this case, it could be something like a channel, a door that is only made to fit a certain molecule. For example, a channel that is made for glucose or simple sugar to go through, then that's the only thing that can go through, nothing else can. But once again, it still has to go with the concentration gradient, from high concentration to low concentration. Osmosis, on the other hand, is a bit different. Osmosis only refers to the movement of water. No solute is moving, only water. Water is moving in and out of a cell. Where water is moving depends on the solute concentration. For example, in this example here on the, let's go back. On the right side here, it looks a bit more concentrated than the left, right? Because it has more solute compared to the left. So water wants to move from the left to the right to dilute the solution on the right. That means the left side would lose some water while the right side would gain some water, since water is moving from the left side to the right side. I want us to take a closer look at osmosis and why should we really care about where the water moves. Here we have a happy red blood cell. This red blood cell is in an isotonic solution. Iso means the same, which means the concentration inside of the cell is the same as the outside. So the volume remains unchanged. Water is not moving. This red blood cell shape remains the same. Now, if we ate a whole bunch of salt, that means the environment around the red blood cell here is saltier than the inside of the cell. This red blood cell is now considered to be in a hypertonic solution. This means the water inside of the red blood cell move outside, trying to dilute the salt. As the cell loses water, it would then shrink and potentially die. On the other hand, if we drank a whole bunch of water, much more than our body needs, something like in a water competition, this means we have a lot more water in the outside environment and makes the inside of the cell more concentrated. This cell is then considered to be in a hypotonic solution. Hypo means below, right? So water from the outside would want to move to the inside of the cell. From outside wanting to go to the inside. Eventually, this would fill the cell with too much water and it would eventually burst and die. You see, too little is not good. Too much of something is not that good either. Even water is good for you, but too much of water would definitely kill you, as your cells would burst to death. Active transport, on the other hand, involve movement of materials against the concentration gradient, meaning things move from low concentration to high concentration. Besides requiring a channel that brings the material across the plasma membrane, it also requires ATP, or energy. One popular example of active transport is the sodium-potassium pump. This brings in 3 sodium to the inside of the cell and 2 potassium to the outside of the cell. This is an important pump that all cells have, mainly to create a concentration gradient for other molecules to go through the plasma membrane. Now, another type of transport is vesicular transport. This type of transport technically still considered as active transport. Vesicular transport involves transport of large particles, large molecules, and fluids across the membrane in a membranous sac called vesicles. There's 2 types of that, exocytosis and endocytosis. Exocytosis refers to things that can be carried in a vesicle, then they get spit out of the cell. Versus endocytosis refers to cell consuming things. This could be consuming a bacteria, for example. In terms of consumption, the cell can eat something that is solid, which we would refer to this as phagocytosis, like phagocytosing a bacteria as a whole, for example. In comparison to that, pinocytosis refers to cell drinking. The cell is uptaking fluid instead of a solid chunk. Anyways, here's an example of a recording of real-life cell trying to eat. Looks pretty cool, right?